USA is waiting for his fixed time. Therefore, we don't want to waste time. Our first speaker is Professor Abdullah Muhammad Al-Saadi. Abdullah Muhammad Al-Saadi is a professor of plant pathology and dean of the College of Agriculture and Marine Sciences at Sultan Qaboos University, Oman. He's received his PhD in plant pathology from the University of Queensland, Australia in 2007. He participated in several strategic missions and national and international committee. Professor Abdullah, the floor is yours.
شكرا دكتور. Thanks دكتور. And good afternoon to everyone. How to move this one? Okay. Firstly, I would like to thank Abu Dhabi Agriculture and Food Safety Authority for inviting me to give this talk, which will be about plant diseases threatening food security in the Arabian Peninsula. As you know, the Arabian Peninsula, the countries in the Arabian Peninsula are Saudi Arabia, it covers Yemen, Oman, United Arab Emirates, Kuwait, Qatar, Bahrain, and also parts of Iraq and Jordan. And most of the area is covered with desert. Most of the Arabian Peninsula is covered with desert. There are some places that are about 3,000 meters above sea level, in which, usually, growers or farmers, they cultivate pomegranates and some temperate crops. So people in the Arabian Peninsula, they depended on agriculture, fisheries, and trade as a main source of income for several centuries. And there are few plants or few crops that are native to the country, like date palms, for example, while many of the other crops have been introduced over the last few years. So new crops have been introduced over the last 5 decades in the Arabian Peninsula. These include different types of vegetable crops like tomatoes, cucumber, lettuce, different ornamental plants, fruit trees as well. But in addition to this, also fertilizers have been introduced, compost, and different products. Now the good thing that we now have diversity in terms of either the vegetable crops or the fruit crops or even the ornamental plants in these countries. But the problem that after we introduce them, several diseases or several pathogens have been introduced also after the introduction of these plants. And many of these pathogens, they started to attack plants or crops that are native to this area. It's really difficult to cover all diseases in the Arabian Peninsula in one talk in less than 1 hour. So I'll just have a case study. I'll focus on plant diseases in Oman and only also some of the plant diseases in Oman, plus some of the neighboring countries. As you know, Oman, we can divide it into 3 areas. The one in the north, it's mainly plains and mountainous areas. And there the temperature ranges from 0, which is at the top of Jabal Akhdar, to sometimes it reaches 50 degrees Celsius in the summer. And usually, in the region, in the northern part of Oman, we grow different types of crops. This is the main agricultural region in Oman. So we grow crops like date palms, acid limes, mangoes, tomatoes, and also we have some temperate crops, especially in the Al-Jabal Akhdar, where we have also pomegranates there, we have apples and different types of temperate crops. In the southern part of the country, because of the monsoon season, is somehow a tropical area. And there you'll find a lot of bananas, coconuts, and different types of crops as well. In the middle part, it's mainly desert, and it's hard to find plants or crops in the middle part of the country. So there is diversity in the climatic conditions in Oman. As a result, there is diversity in the crops that are cultivated, and also there is diversity in terms of the pathogens or the diseases we face in the country. I'll just give now a few examples or some examples on the major diseases in the country, and to start with acid lime, or sometimes called Mexican lime or Omani lime. This used to be number 2 crop in Oman in the 1970s, before about 50 years, before 5 decades. We used to export acid limes or lemons to different places, especially in the Middle East, especially the dried ones, the dried lemons. And the ones you see here, the dried lemons, these are not from Oman. These are from Iran, and they are sold in US market, the UK, and also if you go to Amazon, you'll find them. But what is interesting that you'll find they are called Laymun Omani or Omani lime. This is because Oman was very famous before 5 decades in the export of acid limes, but not anymore now, because a disease came and destroyed the lime industry in the country. So this disease, which is witch's broom disease of lime, or Meknesset Sahira, this disease first appeared in Oman in the 1970s, in the beginning of the 1970s, as you can see in the north of Oman, Shinas and Luwa. Farmers, they started to see, the leaves, they become smaller inside, many leaves are produced, no fruits at all from the parts that have the disease. And within usually 5 years, the acid limes will decline and they will die. There was a visit at that time by 2 scientists from the UK, Waller and Bridge. So they saw the symptoms, they took some samples, they sent them back to the UK. But after doing some investigations and even electron microscopy, they couldn't find any viruses. They started to search for viruses, and they couldn't see any viruses. So they thought that the symptoms might be because of nutritional deficiencies. But unfortunately, the disease spread. After that, it moved from Shinas and Luwa to other areas in Oman, and there was another visit by Bouvet, a French scientist, in the 1980s, and also he came to the UAE in the 1990s. Once he saw the symptoms, he did some studies, and then he found that the disease is caused by phytoplasma, or MLO, mycoplasma-like organism. But it was out of control at that time, because the disease already has spread to different places in Oman. The disease appeared in the United Arab Emirates in 1988. And this is expected because the disease started in Oman, in the northern part of Oman, very close to the UAE. And therefore, it's very natural to see it also in the UAE. So it appeared firstly in Bithna, or Masfout, or Hatta. In 1988, they saw the symptoms there. And later on, the symptoms also were seen in Mleha, Wal Awir. The disease almost within 3 to 4 years, it attacked almost 3,660 trees in these areas out of 99,000 trees that were present, just in these areas. The disease, so after the UAE, it was reported in Iran in 1997, and then after that in India in 1999. So this is the history of this disease. In Oman, it killed almost 1 million lime trees since it was reported before 5 decades. The area under cultivation went down. The production also, it went down. And nowadays we import limes from India, from Thailand, and from different countries after we used to export it in the past. So we did some studies on the disease to find out the causal agent and also what's the problem. The first problem we have is with quarantine. Unfortunately, quarantine wasn't efficient enough in the past before 5 decades. And as a result, many diseases entered into the country. Plus also, sometimes there are new diseases that no one know about sometimes. This one, the witch's broom disease on Lyme, the first time it was reported in the world in Oman, and after that in United Arab Emirates. So once we investigated the pathogens, we tried to investigate the citrus plants that are imported from India, Pakistan, some neighboring countries like Egypt and Lebanon and so on. And many of these seedlings that come to the country or to our country, they have viruses, they have virus, they have different pathogens, and sometimes it's difficult to detect them. You have to have diagnostic tools to detect such diseases. So this is the first problem. So one of the recommendations is to have a strict quarantine, and also to train staff who are working there how to deal with such plants coming from outside or seeds or products. Then we did a study on the acid lime we have in Oman to see if the lime have low or high genetic diversity, but unfortunately the lime we have in Oman has very low genetic diversity, and it's very well known in any crop plants and so on. If they have low level of genetic diversity, it means it's easy for diseases to just destroy them, or for disease to have outbreak of diseases. So this is the other problem also we have. We have limes that have low level of genetic diversity. So the recommendation here is to widen the genetic base of limes, either through different tools. We did some studies also on the vectors, how the disease is spread from one area to the other or from plant to the other. And it's very clear that spread is mainly of two or three things. The first one, propagation. Farmers, they propagate acid limes usually by layering. They make cuttings. So the tree has the disease. Sometimes it doesn't develop symptoms, but it has the pathogen inside it. So they make cuttings, they start to, and this is what happened also when it moved from Shinas, the first place in which it was reported in 1970s, to other places in Oman and later on to the UAE. So propagation is one, and the other thing, insects. There are insects that carry the pathogen, Hegemonous species mainly, leaf hoppers. So these insects, they carry the pathogen, but the problem also, these symptoms, which are small leaves and yellow leaves, they usually attract insects. So it's very important for farmers or growers to get rid of all these. So this is one of the management strategies also, not to stop the disease, but at least to reduce it. It's important to prune all the leaves that are small, that have the symptoms. Otherwise, these leaves will attract more insects, they will have more phytoplasma, and then the disease will progress faster. Also, we try to find out, is there any differences in the symptoms of the disease between different locations? And we did this study in collaboration also with some colleagues from the UAE. So we did it in Oman and also in the UAE. And as you can see that the symptoms are expressed, or you can see the symptoms in the northern part of the country very clear. But if you come to the middle, usually you will find decline symptoms. And you will not find the typical symptoms of the disease. If you go to the south, where we have the moon season, symptoms are really, they don't develop well there. So this is one of the recommendations, to choose an area in the country in which symptoms, they don't develop well, and then start to cultivate limes in these areas. Also, we had collaboration with Brazilians, and they also have limes there. Symptoms, they don't appear in Brazil. Also in India, I know that symptoms appear, but the disease is not as severe as we have it here in Oman and in the UAE. So it's very important to go for such regions. This is also the study we did in collaboration with some colleagues from the UAE, and the same hypothesis. So those that are in the desert, they don't develop the typical symptoms, and the same with the southern part of Oman. We did a study also on the minerals in acid lime, and it was very clear that there is fluctuation in the minerals in acid limes. as a result of the disease, so it's important for growers to give enough fertilizers. Also, the other problem we face is nurseries, private nurseries. When we went to these nurseries, they have, yes, they sell different types of crops, plants, and so on, but they are not certified. So they spread usually diseases, different types of pathogens, different types of diseases. Also, we did a genetic diversity study on the pathogen itself, not the plant, the pathogen now, and we compare the ones we have in Oman with the UAE, with Iran, all of them exactly the same. So if we can develop a management strategy, usually this will work for the different countries as long as the conditions are similar. Then the disease, phytoplasma also, the causal agent, or similar one, it was reported in date palm recently before 2 years in Oman. As you can see, it causes yellowing in Oman. The same also on some of the palms, other palms like this one also, again also. It was reported also on alfalfa, sesame, chickpeas, carrot, eggplant, spinach, field pea, faba bean, rocket, and so on. These are different crops. Just over the last 5 years, we found phytoplasma causing different diseases on these crops, squash and tomato, cactus also. Sorry, I'm just trying to be fast because I think someone else will give a talk after me. Also cycas species, and again you can see the malformation in the leaves because of phytoplasmas. So, and the African marigold, you can see also the symptoms, phyllogy and virescence, the flowers. Usually it it changes the shape or the color of the flowers. So in brief about these phytoplasmas, they really pose a threat to crops in Oman and in different places here in the Arabian Peninsula. There is also we did some studies on viral disease, but I'm not going to take long of, or much of your time. For example, TYLCV virus that attacks tomatoes, and we kept reporting different types of viruses on tomatoes and on different vegetable crops. And the problem with these viruses is that they evolve very rapidly because of recombination. So they recombine like the coronavirus. So it's easy for them to recombine. And one example here is given to you, 3 different viruses, a cotton virus, cassava virus that attacks cassava, and okra virus. Those together, they are combined together and they produce a new virus that started to attack tomatoes. So even, viruses that have nothing to do with tomatoes, they can come together and then infect a new host. We did some studies also how to manage these viruses through agric cover, usually for about 6 to 7 weeks. We also developed in our labs there transgenic plants through genetic engineering, plants that have, transgenic plants that have resistance to the disease, and they proved to be resistant to this disease in tomatoes. There are different types of viruses also, like citrus rosacea virus. It's there, it causes stunting, and also it can cause rapid decline. There are different types of, these are fungal diseases. And the problem also, again, with these products that are imported from outside, like the fertilizers, the compost, the problem they have, you can see the full list of pathogens or different fungi coming just in a single or 2 different types of compost. And some of them are pathogens, they cause diseases in plants. So, and they come, even they come with certificates that they are free of pathogens, free of nematodes, but unfortunately they are not free. We did studies also on the causal agents of different diseases at different stages of cucumber. For example, you can see in the beginning, there is only one causal agent, but later on, towards the end, there are more than one causal agent. And this helped us also to decide on what, to use, what fungicide to use or how to control these diseases. Also, there was a failure one time in the efficacy of some of the fungicides that are used to manage these diseases, and we found out that it's because mainly of resistance. So some of the fungal pathogens, they started to develop resistance to these fungicides. Plus the second thing, there is also fast or rapid biodegradation of fungicides in the soil. So if soil is used for a long period of time, many organisms, microorganisms, they accumulate in the soil and they start to degrade the fungicides. Salinity also is another problem, almost everywhere. And the problem salinity, it can affect crops more than affecting the pathogens themselves. So pathogens, they have tolerance or resistance to salinity. We did also studies on the biocontrol agents from our country. I mean, we tried to come up with biocontrol agents that can manage plant disease. As you can see the studies here. They cause inhibition of pathogens. And this is under electron microscopy. It's very clear. This is the work of some of our PhD students. You can see that they inhibit or they attack the mycelium and destroy mycelium of fungi. And in addition to attacking the pathogens, these biocontrol agents, they also promote growth of plants, so they are helpful to the plants themselves. And this is a long list of about 28 publications just over the last 3 years on this topic from our lab. Mango wilt, Doctor, since that you are in a hurry. Mango wilt also is very serious in Oman. It started in 1998. You can see the rapid decline of mangoes. The disease started again in Oman, in Barka, in 1998. After that, it spread everywhere, and because UAE also, they are our neighbors.
It's also mycoplasma?
Yes, I'm going to finish.
I mean that the disease in mango caused by mycoplasma again?
Yes, you have it here. You have it in Emirates.
Mycoplasma, right? Caused by mycoplasma?
No, this is one caused by a fungus, ceratocystis. So this one is a fungal disease also. So it started in Oman, then appeared in the United Arab Emirates, then Pakistan, and before that, Brazil. It destroyed more than 200,000 mango trees in Oman within 5 years, and several also ones in the UAE, especially in Fujairah and the eastern part of United Arab Emirates. There are other diseases on date palms. There is no time to talk about them. On citrus also, on mango. But just the recommendations from this is that we should have quarantine, efficient quarantine in our countries. Otherwise, diseases will be, pathogens, they will be introduced in our countries. Then it will be difficult to manage these diseases. And all these ones, the one on acid lion, for example, the one on mango, the others also, the viruses from Burkina Faso, from different parts of the world, they come to Oman and they come to our countries, and then they evolve, and then they start to attack new crops. Educate growers also is important for us to educate them, train our staff who are working in quarantine and different places, also regular monitoring of diseases and surveys in the country, and also the development of integrated disease management strategies. If you need more details, you can go to these websites. They will have about 250 publications on diseases. I would like to thank our bachelor, master, and PhD students who did most of this work and our supervision. I'd like to thank also our colleagues, research assistants and staff, also collaborators from Oman and from different countries. And one of them is here, Dr. Mohammed, who collaborated with us in the past also. So thanks to all of you. I'm sorry to be short.
Thank you so much, Professor Abdullah. Really a very interesting presentation. I'm really excited about the huge work that you have done in Sultan Qaboos University, and in particular, the big number of publications coming out of this research. This reflects the deep science you have in your department at Sultan Qaboos University. Congratulations. Okay, now the floor is open for questions. Before that, I would like to welcome our Director General, Director General, His Excellency Al-Bahri, and his team, also a high administrator, VIB people. You are welcome. And yes, some question, if possible. Tariq, you have a chance. I will give a chance for after. Yeah, please. Then Dr. Salama, and then followed by Tariq.
Ishraq Jafar Ibrahim from Sudan. Thank you for your interesting presentation. I have a question. You use a biological control for treatment of the disease. What about the side effect of this biological control on human?
What is it? Sorry, I couldn't hear you.
You use a biological control for treatment. What is the effect of this biological control on human who consumes this plant? Okay.
Thanks. Yes, as I said before that we did some work on the biological control of plant diseases, and just over the last 3 years, 28 papers were published on this issue. Her question is about, is it going to affect consumers? Usually, these biocontrol agents are applied to the soil. So most of these biocontrol agents that have been tested, they are mainly for soil-borne pathogens that are in the soil. So they don't go usually to the fruits, unless if it is a fruity crop. And most of them, see here, before we release any biocontrol agent, usually it has to go through safety check. So we have to make sure that it's safe for humans. It won't cause anything to humans. Yeah.
Yes, Dr. Salama.
Shukran, Doctor. Thank you very much for the valuable information that you have provided. My question is, you have really rich scientific outcomes, research. How we can bridge the gap between the research, the science, and the policymaker? Because I can see from the research you have published, there are many outcomes that could go to policy, to procedures, and to be implemented even by the farmers, engaging the farmers, awareness campaigns. So a lot of your research really need to be translated into into action for policymakers and others. So what do you see the challenge, and how we can bridge the gap between the science and the policymakers? Thank you.
Okay, thank you, Doctora. Okay, this is a difficult question. See, Doctora, usually all the work that we do here, and you can see that about 250 publications just on plant diseases, most of the work has been done by our students. And our students, usually they come from the Ministry of Agriculture, the Royal Court Affairs, and so on. So usually we bring them there, we train them, they do the work, and then after that they go back to their work, the supervisors, and so on, and they start utilizing research outcome in their places. Plus also we have in Oman regular meetings, but that was before the COVID-19, we stopped. Regular conference like the one you have here, usually it's jointly with the Ministry of Agriculture, and we usually present research outcome every year to the industry. Also, many of the research we do, at the end we usually write reports and we send them directly to the Ministry of Agriculture, Royal Court Affairs, and different stakeholders. But still we need to do more really, in this regard.
We have time for just one question, if no more questions. Yeah, okay, please.
Thanks, it's covered by Dr. Salam.
Okay, thank you very much. Professor Saadi, thank you so much for this very interesting presentation. Hope that in future we collaborate regarding these very dangerous and threatening plant diseases. Thank you very much.
Thank you. Thank you.
Okay, now our next speaker is Dr. Tahir Rashid from Alcorn State University, USA. So I'm waiting for the technical team to connect us with Dr. Rashid. Not coming? After we we have a coffee break. And then the next session. So if you let me make a change, we can skip or postpone the presentation by Dr. Rashid Tahir and jump to the next session. If you do agree, then is Dr. The first presentation, Dr. Rehab Yassin. Are you ready? Sorry for this shift. We are obliged to do so because of the problem with the connection. Hope that Dr. Tahir Rashid will join us in the coming few minutes. Now we are going to listen to Dr. Rehab Yassin Gharib from Arid Land Cultivation Research Institute.
From
in
City of
Egypt.
Scientific Research and Technological Application, برج العرب Alexandria, Egypt.
Thank you for correction.
Thank you. السلام عليكم جميعا. Utilization of Cladophora glomeratta extract nanoparticles as eco-nematocidal and enhancing the defense response of tomato plant infected by Mildew javanica. The first, tomato plant as important vegetables group that belongs to family Solanaceae. Root rot nematode, especially Mildew javanica, Arenaria, and incognita, is the damaging genera of phytoparasitic nematodes on tomato. Nowadays, the biological synthesis of metallic nanoparticles are numerous benefits of eco-friendly for pesticide purpose due to the absence of toxic chemical synthesis. Cladophora glomeratta is a freshwater macroalgae. Is the very interesting example for green synthesized nanoparticles. Materials, Mildew javanica and seed tomato. Examine that. We synthesize green nanoparticles from Cladophora glomeratta extract and reduction of AgNO3 by bio nanoparticles. To examine the activity of green synthesized silver nanoparticles against nematode Mildew javanica, we found in the laboratory experiment with compare the commercial nematicide. We found when compared with the commercial nematicide, rugby nematicide commercial, the reduction percentage egg hatchability of Mildew javanica about 18-70% after 20 hours with 100 concentration and 100 after 24 hours from exposure time. And when apply for the mortality of larvae on second juveniles mortality, we found 19% concentration on juveniles. We also examined this mixture against nematode in greenhouse. We found the highest growth parameter in the fresh and dry root and shoot system. We found reducing the gall and egg with 19%. Real-time PCR is comparative technique to quantitative the expression level of the defense gene like phenylalanine ammonia lyase, PAL, and polyphenol oxidase, and finally peroxidase. We found that The green science nanoparticles in the expression gene of peroxidase here. In peroxidase and BBO and BAL is a high expression level after 14 and 28 days after treatment. This data suggests the fundamental role of green synthesized nanoparticle on activation, upregulation of defense-related gene, and the induction the defense against infection of root rot nematode. We suggest that the green synthesized nanoparticle can be strongly nominated as an eco-friendly nematicide against root rot nematode. Thank you.
Thank you very much, Dr. Rehab, and thank again for being in time. So we have time for questions. Okay, then thank you again so much, Dr. Rehab, for this very informative presentation. Now we have like 20 minutes before the coffee break. Would you like to continue listening to one presentation and then we move to the right? Correct? Do you agree? Okay. Is Dr. Vishnu Baharat is with us right now from Khalifa University? Okay, then we have also Dr. Ibtisam Alabri. Ibtisam is presenting virtually. Okay, then we have Abdul Malik Khalafallah. Is Dr. Abdul Malik is there? Yes. Okay. Okay, Abdul Malik Khalafallah is a professor of veterinary virology. He's currently working at AFSA and in the veterinary laboratories. I just want to talk a little bit so that they are ready for my presentation. Abdul Malik Khalafallah. Yeah, okay. Okay, now join me to welcome Dr. Abdul Malik Khalafallah. Okay, my presentation is about, I have two microphones. So my presentation is about One Health's contributions towards effective responses to coronavirus infections at AFSA experience. Coronaviruses are diverse viruses, but we are going to focus on the MERS coronavirus and SARS coronavirus 2 that cause the COVID-19. Okay, I'm going to focus on what do we mean by One Health. One Health program components and ADAFSA and One Health, and then ADAFSA experience on testing for coronaviruses and lesson learned. I don't think I need to define the term One Health, but the most important thing is the component of the One Health approach. What I mean, the three pillars, animal, human, and the environment. According to OIE, One Health concept ensure political support in coordinated prevention of high public health and animal impact diseases at the human-animal interface. But as you saw in the previous presentation by Professor Saadi, I think plant diseases are also or should be involved. Unfortunately, plant disease has no organization like OIE. So I suggest that to have one, and I propose you to lead this organization, which could be named OIE is World Animal Health Organization. So it would be World Plant Health Organization. Okay. The principle is of One Health have now been taken up and are applying them with, sorry, excuse me. Okay. The principle of One Health have now been taken up, and it's now on the political agenda, particularly after the MERS corona and SARS coronavirus 2 infection, the COVID-19. Food safety at the center is now at the center of One Health approach for combating zoonosis. One Health also has relation to socio-ecological aspects, and the perspectives are just ensuring social and cultural dimensions. You know about this, what they call it tripartite collaboration, which means the three involved organizations, the World Organization for Animal Health, OIE, and WHO, as well as FAO, Food and Agriculture Organization. They should work together to achieve the objectives of the One Health. The element of One Health program usually contain elements of field epidemiology related to animal diseases, and then sharing resources of laboratory health information, and then the use of multi-sectorial team from the vet labs, vet services, and from the human health. These teams should be working together to achieve the One Health approach. Then application of an evaluation framework, coordination mechanism between the veterinary and the public health sector. Here I'm going to show you how we fulfill the requirement for animal health, for the One Health in Abu Dhabi in particular. This is a map of United Arab Emirates with Abu Dhabi in yellow. And generally speaking, UAE has set national bodies within or supported by the government to provide One Health approach to zoonosis and emerging infectious diseases since 2015. There are a number of bodies that are participating in the One Health, but here I just want to concentrate on two parts. The Department of Health, the human side, and ADAFSA, the animal side. Of course, there is like, for example, the environment sector has also something to do with regard to One Health. Like, for example, the destruction or removal of carcasses, other aspects, they are involved. But with regard to coronaviruses, we are just concentrating on Department of Health and ADAFSA. And as an umbrella, we have what we call it NCEMA, the National Emergency Crisis and Disaster Management Authority. This is a federal organization that govern or manage all the activities. So they are just like an umbrella that cover the activities. We are this organization has experience with regard to some zoonotic pathogens like MERS corona and Crimean Congo hemorrhagic fever virus, as well as anthrax, rabies, and brucellosis. So the way how these parties work together is something bidirectional. I mean, from human health sector, like notification of a disease, to the animal health sector, and vice versa, from the animal health sector to human health sector, and down integrated response, like actions to do field investigation on animal diseases, and also to do field vaccination on human diseases. The key enablers include early warning response system and traceability of suspected animals through animal registration system, and then emergency response plan, as well as laboratory capacity and continuous communication and information sharing. Here I just want to show you the number of samples that were received at our laboratory, the veterinary laboratory division of ADAFSA, based here in Abu Dhabi. With regard to MERS coronavirus detection, we started early since 2014, and we did rabbit test ICA, as well as reverse transcriptase qPCR. So if you look at the number were very huge in 2014, like 34,000 in total, and then declining until we worked until 2021, in which we have only very few number. The total number tested of sample is 41 and 643 samples. Just imagine how huge number of samples that has been tested at our laboratory. Besides the testing for diagnosis, we also have done virus isolation. We have done isolation of the virus, and now we have 5 isolates from camels, and one of them was besides 100 times in cell culture. The purpose is to look at the mutations that could happen during this passage in cell culture, as well as to prepare for a vaccine. We have a plan for producing a live vaccine and kill vaccine in order to protect human. As you know, the disease in camel is not a well-defined disease. There is no coronavirus disease in camels. If you go to the field, ask a veterinarian or ask a owner, have you seen any case of MERS corona in your camel? He will say no, never. Because the disease causes no overt clinical signs. But the purpose of preparing for vaccination is to inoculate the camel in order to eliminate the virus and protect human from getting infected from the camels. And this is very, very important, especially with regard to the very close relationship between the camels and the owners. Owners right now, they don't believe that their camels are the cause of transmission of the disease to human. Then with regard to SARS coronavirus 2, which caused COVID-19, we also tested a number of human samples. If you look at the table on your left side, you will see that we started in March 2020. At that time, when the Department of Health received a huge number of samples, there was no way out but to contact us, asking us to help. Bearing in mind that we have a previous experience with coronavirus, the MERS coronavirus. We have the facilities, we have the technology, we just need to change our setting to introduce the biosafety level 3 and to change the primer or the PCR condition and continue our normal routine. This is why they call us to be involved in the testing of human samples. So, but it started from March 2020 until February this year, and the total sample tested is around 80,000. We have also done another testing for animal samples for SARS coronavirus, because there is an incrimination that this disease could be brought to human from animals. So we have done the research targeting animals in contact with humans. So we tested camels, cattle, sheep, goats, dogs, cats. I have the number. As you can see, total we have like 1,020 samples from human. Unfortunately, all were negative. I don't want to go deep into details. I tell you what are the results. Negative is not beyond, this is beyond my speech. I just want to show you how we have done a very good work with regard to coronaviruses and participation in One Health. So we also, according or based on a request from the ministry, from the environment side, to perform a study on environmental samples. So we started in January 2021, and at the end, in August, we have done testing 2,419 samples. The sample included sample from the supermarkets in Abu Dhabi, imported food consignment from the border, airport, and seaport, also the knives and working surfaces of the abattoir, poultry, slaughterhouse, also fresh meat, fish, vegetables, fruits, markets, and also from food handlers. Here I can say that there is some positive. Besides the testing, we have also done sequencing of 6 SARS coronavirus genomes. And the result, the sequence are now deposited in GIS AID database under accession numbers, as you can see. The question will be at this point is how we have done this huge testing. So we have a number of extraction system, automated extraction system. On your right side, here you have this, they call it TACO, and here this is from Kaijin, EZ. Each one can do testing 14 samples, and this is an automated system, Kaya Symphony. And so we have a number of these extraction systems that allowed us to test and like even up to 5,000 samples per day. Dr. Hassan, correct me if I'm mistaken. Additionally, we have also some equipment that are being used for sequencing. These are all for the QuantiStudio and Rocha, Biorad. These are all for partial genome sequencing. I need someone to say time, but I'm managing the time, so don't worry. There is no problem. or there is a problem? Okay. These are, look at the whole lab is full of these extraction and machines. We have this system, we call it pocket, all in. So this can do extraction followed by sequencing. Followed by PCR. So it's just you put the samples, and then you are you go walk away and come back to just to record the result. Each machine can do 8 samples in 1.5 hour. We have like 30 units of this, 30 working at the same time. But just imagine how our team, our staff is just busy with going from here to here until just to adjust here, and sometimes there is some problems in one machine or the other. Here we show you the technology of genome sequencing. We have what we call it Minayon nanoboard system on your left side. This one, very small machine, but very, very, very effective. We have also Illumina MySeq for whole genome sequencing, and Sec Studio for partial genome sequencing. These machines allowed us, as I told you, to sequence 6 SARS coronavirus genomes detected from the environmental sample linked to positive cases. And as I said before, they are now deposited in the gene bank. Okay, at the end, what are the lessons learned from implementing the One Health from this contribution from the veterinary laboratory, from the animal health side? Adaptive laboratories were considered among the first veterinary laboratories in the world to have managed to test human samples. We are not alone. I think we have the Italian laboratories also involved, laboratories in Portugal also involved, in USA also involved. So we are not alone, but we are among these laboratories, veterinary laboratories that were involved in testing human sample. The experience gained in managing MERS corona virus and testing for SARS coronaviruses have provided ADAFSA veterinary laboratories with insight into ways to address the COVID-19 pandemic and prevent future spillover, because we are, as I told you, we are searching for the origin of the virus in the environment or in animal commingling with human. We are doing this research continuously to just address the issue of the origin of the virus. Our result point to persistence of SARS coronavirus 2 on fomites, which is potential biosecurity concern for humans. Fomites, I mean, as I told you, we have 6 positive cases from one, I think, from a bakery. Hassan, you can correct me, from bakery. We collected sample swabs, and this swab tested positive. So meaning that there is environmental issue with regard to SARS coronavirus 2 that should be taken seriously. Our result point to persistent veterinarians' experience and knowledge can improve the One Health approach to emerging and zoonotic diseases. And as part of multidisciplinary teams, veterinarians should continue to engage within the One Health framework. And that's all. Thank you very much. So I have to ask you to raise questions, comments. Yes.
Firstly, thank you very much for that interesting presentation. I think it's quite amazing to see how the number of samples tested was increased so rapidly during the pandemic. And certainly at the OIE, we were very appreciative of the Abu Dhabi Agriculture and Food Safety Authority to developing guidelines for other veterinary laboratories to test SARS-CoV-2 specimens. I just have two questions. The first one, do you feel the participation of ADAFSA in this work of testing human specimens has raised the profile of veterinary services across government, you could say? And the second question, based on your experience in the pandemic, do you feel you're prepared for disease X, for the next big emergency that could happen? Thank you.
Okay, thank you very much for your question. Yes, with regard to the first question, is ADAFSA. The experience of ADAFSA in testing coronavirus has raised the profile of veterinarian in the, if you mean globally or internationally, I say yes, indeed. After that, as a result of this contribution, we received so many consultation requests. They consulted us, OIE also consulted us. Additionally, the FAO also contacted us, and now we are preparing to develop a collaborative research with FAO. So internationally, there is the recognition for our contribution to the One Health. Internally, yes, for sure, we receive a number of acknowledgement from internal bodies like the ministry and higher authorities. So I assure you that there is recognition of this contribution. And we are proud in the veterinary laboratories of ADAFSA for what we have done with regard to the One Health. There is a lot of success story I had no time to talk about, but I tell you something about the MERS corona. We have published a number of research articles on the contribution of ADAFSA with regard to One Health. And I remember in one international organization, people are saying, frankly, we are proud of your experience. The implementation of One Health in Abu Dhabi was very fantastic. The second question, sorry, sorry, can you repeat, Daniel?
About disease X, so the next emergent disease.
Okay, this is a very interesting question. Yes, I can say yes, but I'm not sure. For disease X, you know, the disease X is expected to be within the family, I'm talking about virology, family coronaviridi. Very few, very few scientists point to other like influenza viruses. But influenza virus is a traditional virus. So people are talking about the disease X will be within the family coronavirus. Therefore, we are now developing or in cooperation with FAO to have a system of RT-PCR using primers that what we call it generic primers or PCR systems that can detect all or expected coronaviruses. And we are developing this because there is not a validated test We are going to select the best protocol and to collect sample from the environment, from the animals around human, and test for this pan-coronavirus PCR. Once we get something, we will continue sequencing. It might be this disease, and if we detect it early before jumping to human, this will help a lot in tackling the issue of the disease X. Satisfied? Yes, Tariq? After this, we'll go for a break.
Thanks,
Yeah, please.
Prof. Abdul Malik, for your presentation. My question is a very short question. I forgive your abusing the time control. Anyway, I mean, Adafka has a very outstanding experience in packing up the public health laboratories during the past COVID. But this experience has not been expressed quantitatively. That means, then people spoke about the good role, or the very supportive role played by ADAPSA laboratories in packing up and playing a major role in combating the crisis. But this effort needs to be, light need to be shed upon this experience in quantitative fashion. That means how, because the public understands this, how much you contribute to solve certain problem, and how much this is to test the real contribution size and impact.
Yes, thank you very much. If I understood your question, you are talking about these achievements are not, conveyed in the right way to the public. Well, I can say that we are not politicians. We are not. We don't want to shed high level of light on us because it will cause blindness. Just we'll leave things go, and people one day they will acknowledge our contribution. Is that okay for you? Yeah, we're not showers, and we want to, whenever there is a place, we want to show that we are the best. We are not. As I told you before, we are just participating in the One Health approach. Whether we are the best or something else is let other people judge on this issue. Okay, by this I think we are now. It is time for a coffee break. Let us just because we are now one family. Let us look at the agenda in case we think go smoothly. We can finish just before the allocated time. We have just a minute. We have 20 minutes for the break. If you allow me to suggest something, that I think 10 minutes is okay. Fine with you. Just you go and drink some coffee, then come back in 10 minutes. In this case, we'll finish a little bit earlier. What do you think? You are free to say, no, I need 20 minutes. Sorry? Yes? Okay, let me. Yeah, okay, let me see. We have one virtual presentation that we missed from Tahir Rashid, USA. This is one. We have done with Rehab and Abdul Malik. We have one, Niveen Rami is here. Niveen, you are, okay. And Yusuf Olos. from Robert Biological System. In person, I think. It's not, we need to be sure that he is here with us. Yeah, he's here. So now we have to, Vishnu didn't appear. Vishnu Bahadran. Is Vishnu Bahadran from Khalifa University with us or not? So that means we have only two in-person presentations as well as one virtual. Okay, fine? No more than 40 minutes or so. Okay, let us fix the time. So at 4 and 11, 4:11 come to rejoin. Okay? Good. Sorry? You have 10 minutes. Yeah, please.